Oh, what is it? Staring at us with such anguish face. It's a very dangerously looking creature. Is it an organism? Is it a non-living thing? What is it? Oh, it's saying by itself only. It's a virus. So we are going to discuss about the details of a virus. Oh, why the boy is scared? Let us look. Oh, it's scared about that of a virus which is roaming out. As because he thinks that the virus, it is just poison. Why? Because the virus word itself says it has been derived from Latin, which means a poisonous fluid or a venom. Now, who are these great persons? Oh, they are the one who had contributed a lot in the virology. The great Ivanovsky, sir. He was the first one to recognize submicroscopic particles as the main causal agent for the tobacco mosaic disease. The Beijerinck, the first person who had referred the fluid as the contagium vivum fluidum, which means the living contagious fluid. And the great Stanley, sir. He was the one who had first of all crystallized TMV for the first time and let us know that the viruses, they can be crystallized under in vitro condition and can be studied also. Who is that mysterious creature? What makes this creature a mysterious one? Well, a virus is actually an ultramicroscopic. That stands for it is so much tiny or so much minute that it can easily pass through a bacterial filter. A virus is a acellular organism. Acellular means it is not having any kind of cellular structure. It cannot multiply on artificially synthetic medium. That means either in agar medium or any other kind of medium, it cannot multiply. It always requires a living host cell condition for its own multiplication and growth. The most importantly, it is metabolically inert. It is an inactive particle which is outside. That means it is not showing any kind of metabolism, any kind of growth, multiplication feature when it is placed outside of the host body. But as soon as it gets inside the host body, it becomes active and it shows its active fast rate of multiplication. It's an obligate intracellular parasite. What does it stand for? Obligate stands for strict. That means all the viruses, they always require a host cell compulsorily for their multiplication. And they do not live only outside the cell. They gets inside the cell with the help of their own genetic machineries and they shows their multiplication process. What more feature makes virus a mysterious identity? It never grows in size. Although it multiplies, but it never shows any kind of growth. It always shows host specificity. Means a plant virus, it will attack an animal and the vice versa. A very typical system which we are referring it as Lippmann system, which actually tells us that how a cell, it synthesizes ATP by utilizing its own machinery. It is never present within that of a virus. It's highly pathogenic and infective. Whenever it gets inside that of its host, it causes a severe form of damage. It can get easily transmitted from one host to another via any kind of means. It can be via water, it can be via food, it can be via air, it can be via mere touch. Most importantly, it shows heritable form of mutations. Means it changes the cell's genome in such a manner that it brings a drastic change within the body, and sometimes those changes they can get heritable also. Oh, the virus here it is confused. Why? Because it thinks that it resembles with a non-living object. But why is it thinking so? As because it lacks a protoplasm, it is metabolically inert or inactive external to the host cell. It shows an absence of energy synthesizing as well as storing system, as well as it shows a complete absence of any kind of metabolism. It can get crystallized and it can never show growth. Now again the virus is confused. It thinks itself like a living object. But what are the features of it that resembles like a living object? It shows the presence of a genetic material. It can be either DNA or RNA, but not the both. It possesses the ability to multiply within the host cell. And it can undergo mutation, that is, the sudden change in the genome. It can get easily killed by autoclaving and ultraviolet rays, like the other cells also. It is made up of many different kind of organic macromolecules, including a very important one, the proteins, as well as the nucleic acid. How a virus is classified? Holmes in 1948 given a classification of virus on the basis of the host specificity. A virus, it can be classified on the basis of the host in which it penetrates, it multiplies and takes a living abode. Like there can be few viruses which takes the shelter within that of a plant cell. We are referring these viruses as phytophaginae. The phaginae word means eating or attack. Most of the phytophaginae consist single-stranded RNA as a genetical material. For example, TMV, tobacco mosaic virus, PMV, potato mosaic virus, etc. There can be few viruses which acts over the animal cell. They attacks the animal cell and we are referring them as the zoophaginae. Now, within zoophaginae, there can be more than 50% zoophaginae viruses which contains DNA as a genetical material. For example, pox virus, herpes virus, etc. And in less than 50% cases, there can be RNA as that of the genetical material. For example, reoviruses, measles viruses, chicken pox, like AIDS viruses, which is a retrovirus, etc. There can be few other viruses which attacks a bacterial cell. We are referring them as the bacteriophaginae. For example, T. phages, lambda phages, etc. They can consist of either DNA or RNA as a genetical material. Viruses are extremely minute and can be easily filterable through the bacteria-proof filters. So what is the size range of a virus? A virus size, it ranges from approximately 10 to 300 nanometer. The smallest plant virus, we are referring it by the record as satellite tobacco necrosis virus, which is having an approximate dimension of 17 nanometer. And the smallest animal virus, we are referring it as the cattle's foot and mouth disease, as well as rhinovirus, which is of approximately 10 nanometer in its dimension. There can be many viruses which can have a larger structure. Among many plant large viruses, one of them are tobacco mosaic virus, which is having a dimension of approximately 300 by 17.5 nanometer. While in animal, the largest one, it can be the pox virus among many, which is having a dimension of 230 upon 300 nanometer. What is the shape of a virus? We have to remember that an intact, inert, complete virus particle which is capable of infecting the host and is lying outside the host cell in cell-free environment is referred as a virion. The capsomeres in the protein coat, they are arranged in order to give definite shape to the virus particle. A virus particle can show three major kind of symmetries, as it can show a helical kind of symmetry where capsomeres are arranged in a spiral manner like in TMV. It can show a cubical kind of symmetry in which the body of virus is spherical or polyhedral in shape and the capsomeres are arranged in the polyhedral manner. It can also arrange itself into a special kind of symmetry which is referred as a binal or complex symmetry. In this case, the viruses can have a combination of both cubical and the helical symmetry. For example, in case of pox viruses. The fine structural component of a virus is actually composed of two major compositions. The first one is a capsid, which is actually an external proteinaceous protective coat and determines the host specificity of a virus. It actually forms 95% part of the virion. It's huge, isn't it? It consists of specific proteins to protect the nucleoid, that is the internally located genome, from nuclease enzyme digestion. It has antigenic properties and is composed of many identical subunits called the capsomeres. Thus, a capsomere is actually a single polypeptide of few amino acids. For example, in TMV, it is consisting of approximately 158 amino acids. The internally located genomic structure is referred as nucleoid, which is actually a single copy of genome, can be either DNA or RNA. Thus, the viruses are haploid except few like retroviruses, which are showing diploid as they contain two identical single strands of RNA genome. Now, few animal viruses like influenza virus, arboviruses, rhabdoviruses, etc. can have an external outer covering which is referred as the envelope. Those viruses thus which are not having the envelope are referred as naked, and the viruses which are having the envelope, we are referring it as enveloped virus. The composition of an envelope is actually the lipid and the carbohydrate as well as proteins. The protein part with carbohydrate, it forms the glycoprotein. The lipid and the carbohydrate part of the envelope, it is actually derived from the host cell membrane during the exit of the virus from the host cell, while the protein is coded by the viruses itself. For example, retroviruses like HIV and others are always enveloped. A virus reproduces in two major ways: the phagic mode of reproduction and the pinocytic mode of reproduction. A phagic mode of reproduction is exhibited by different kind of phaginoviruses, while a pinocytic mode of reproduction is exhibited by viruses like tobacco mosaic virus, HIV, hepatitis B, etc. In a phagic mode of reproduction, the bacteriophage it undergoes two kind of stages. In first kind of stage, a bacteriophage kills the bacteria which they infect and release the daughter viruses after rupturing. These kind of the phages are referred as virulent phages, and they follow a typical lytic reproductive cycle for continuing their reproduction. While the second stage of virus, we are referring it as the non-virulent or temperate phages, which have the potential to kill the host but normally do not lyse or kill it and involve the integration of viral genome with host DNA to form prophage. The lysogenic cycle is actually followed by the temperate non-virulent phage like the lambda phage, while the lytic cycle is actually followed by that of T series phages like T2, T4, etc. The lytic cycle of the viral reproduction is mainly observed in T series of bacteriophage like the T2 and the T4, and is first reported by Delbruck, Hershey and Luria. The lytic cycle involves the first step as the adsorption of the T2 and the T4 phage to specific receptors on the cell wall of the bacterial host with the help of its tail fibers. The lysozyme present in the tip of the tail, it makes a hole in the cell wall of the bacterium. Then the tail sheath contracts and the phage, it injects its DNA into the bacterium. This causes the virus an approximate of 140 ATP molecules. The capsid and the tail sheath, it remains outside the cell, and they are referred now as the host body. This viral DNA takes over the host machinery and transcribes an early mRNA to form specific proteins for making new enzymes like nucleases, which will degrade or dissolute bacterial or the host DNA and the RNA. The viral DNA now will direct the synthesis of its own type of DNA and late proteins which will form the capsids, and all the parts will get synthesized separately. Gradually, after all these steps, the viral components now will get assembled one by one. First of all the head, then the neck, then the tail, and finally the tail fibers will get added in the last to form the new phage. As soon as they are getting assembled, the lysozyme, it will lyse the bacterial cell to release about 200 new phages, and the number of virions produced per host cell is specific and will referred as the burst size. The next cycle of viral reproduction is referred as the lysogenic cycle, which is mainly evident in non-virulent phages like the temperate phages, example, the lambda series of phages. It was first reported by Lyoff and Woolman, and it involves the first step as adsorption of the phage on the host cell surface by the contraction of the lambda phage tail sheath, after which the lambda phage, it injects its DNA into the host cell. This DNA, rather than that of taking controlling of the cellular machinery of the host, it actually allows the synthesis of a repressor protein which keeps the viral genes in a repressed state. Thus, the phage DNA, it actually undergoes a reduction to that of temperate or non-virulent state. Now the phage lambda DNA, it actually attaches to the specific site of bacterial genome with the help of lambda integrase enzyme. The phage DNA in this latent, that is a non-virulent form, it is referred as the prophage or the provirus, and the bacterium carrying a prophage, we are referring it as the lysogen. The bacterium continues to grow and divide, and the prophage, it replicates as part of the bacterial DNA. Is there any other particle which looks like a virus? Yes, there are certain kind of virus-like particles which actually lacks one of the essential component, either the proteinous capsid or the nucleic acids. They are referred as the subviral particles or the subviral agents. There can be in total 3 types of subviral particles available. First one referred as the viroids, the second one known as the virusoids, and the third one we're referring it as the prions. So, we'll start the discussion of the siblings of a virus, that means the subviral particles, with the discussion of viroids. The viroids are actually the smallest known subviral particles and are also known as the infective nucleic acid entity or the metavirus or the pathogen. They are actually the naked single-stranded RNA molecule of very low molecular weights. These kind of subviral particle, they actually lack any kind of external proteinaceous covering, that is the capsid as well as the envelope, and they are highly contagious, causing diseases only in cultivated higher plants like potato spindle tuber disease, PSTD, etc. These viroids are transmitted by mechanical means, and they are first of all discovered by T.O. Diner. The viroids are actually consisting of short-stranded circular RNA which has inverted base sequences at their ends, and thus they resemble very much to the retroviruses and the transposons, which are also known as the jumping genes. Hey, wait! 2 potatoes are interacting, but one of them is of normal spherical shape, whereas the other is of typical spindle shaped, and the second potato, it looks very sad. Why, what happened? Let's see. The first potato says, "Hey my dear potato friend, why are you so dull and of typical spindle shape?" The second potato says, "What to say my friend, there is a viroid inside me." Oh, now we understood. This is a case of typical potato spindle tuber disease, in which a normal healthy potato, whenever it gets a viroid inside it, it changes its shape to that of a typical dull spindle one. The second subviral particle which we are going to discuss is known as the virusoids or the satellite RNA. What are virusoids actually? These are the viroids which are containing small circular RNA and are located inside the protein coat of a true virus. Why I am referring it as a viroid or are similar to viroid? As because they do not possess any capsid and they are dependent on their host viruses for their replication. They replicate within their host itself and they do not cause any kind of infection. They were discovered by Randles et al. The final type of subviral agent which we are going to discuss is referred as the prions or the slow viruses. The prions are actually the proteinaceous infectious particles which are devoid of their own genetical material. The prions were first of all studied, isolated and identified by Stanley de Prusina as a main causal agent for diseases like scrappy, kuru and mad cow disease in 1982, and he was also awarded the Nobel Prize for prions in 1997. Prions are actually produced by a mutation in the normal gene of the host DNA, which produces an abnormal glycoprotein called the prion protein. that induces the normal glycoprotein to fold incorrectly, causing it to form aggregates of approximately 10 to nanometer in size. They accumulate in the brain cells and slowly and progressively damage as well as destroy the living brain cells. Actually, prions replicate very slowly. It is having an incubation period of approximately 1 to 4 years in the spleen and lymphatic system from where they pass to brain and damage the cells. So, the prions are actually derived from the normal body proteins itself by spontaneous genetical mutations. Hey, wait. I'm saying something surprising over here. What's actually happening? I think a tribal person with a very heavy heart, he is trying to eat the brain of a dead relative. What? But why? Why is it so? Actually, this is a common ritual within the tribes of the New Guinea, in which the individuals they used to participate in the cannibalistic consumption of their dead relatives as a mark of respect and mourning. And they used to test the dead brain tissues. The infected persons are called kudu, that is the dancers. The disease finally results into dance of death or the laughing death, as because the individuals who used to consume the brain within their body, there shows an abnormal mutation in the glycoprotein of their brain cells, which results into the formation of the prion proteins, which gradually results into the formation of this disease, which we are referring it as that of the kudu. It's a very deadly kind of disease which were very much prevalent in the tribes of the New Guinea while they used to participate in this cannibalistic kind of consumption of their dead relatives. Now, this cannibalistic ritual has been banned by the New Guinea government itself. In this respect, I would like to add one more thing regarding prion, that the prions are actually resistant towards different kind of enzymes like nucleases. They're also resistant towards heat, proteases, radiations, as well as disinfectants also. Oh, what a nice, cute, smiling bacteriophage. What is it saying? Thank you. Thanks a lot for watching our presentation. If you want to see more, kindly subscribe to our channel. Please press the bell button and like and share. Thank you. Finally, now the time for the credits. So, all the kind of pictures, whatever we had seen till now, especially the illustrations, definitely they've been inspired from various sources, including the online sources, internet sources. But all the illustrations are handmade and they are done by Mr. Soumitra Bhattacharjee, that is myself only. The various printed pictures, like those of the great personalities, the scientists, are directly taken from the online sources, the internet sources itself. Finally, thank you very much for patiently viewing the presentation. And as the cute bacteriophage informed earlier itself, kindly press the bell button as well as subscribe, put the like, so that more such interesting, attractive presentations can be brought about. Thank you.